Hey, how's it going, guys? In today's video, we'll be talking about prokaryotic transcription. And why is this important? This is important because what transcription does is it turns DNA into RNA, right? And this RNA is used as a messenger of information. This is a very important key point. And what it does is it transfers this information to the cytoplasm where proteins can be created through translation, right? So now let's look at prokaryotic transcription, which is different from eukaryotic. It is the same, but very, very simpler, actually. Eukaryotic transcription takes all these different binding proteins and transcription factors, right? Just a whole mess, right? But prokaryotic transcription, all you need is something called a sigma factor, okay? So let's move on to this next slide. Let me see here. Get it prepped. Okay. So here we have a sigma factor, and what a sigma factor does is, let me just circle it here. That's a sigma factor, and what that is it's a protein needed. Well, it's a protein that's only found in prokaryotes, right? And this is needed to initiate transcription, hence the name initiation, right? All right. So what happens here? The sigma factor binds to the polymerase, this big beige brown, whatever you want to call it, that thing there, right? That's the RNA polymerase. Sigma factor binds to that, right? And this whole, I put DNA complex, but it's supposed to be RNA complex, whatever. This polymerase, it slides along the DNA until a promoter is met, right? So that would be this yellow region here, right? So now, once that complex meets that promoter group, what's going to happen is that sigma factor is going to bind to the promoter, as we see in this step here. Okay? So first we have it binds to the polymerase. Then after that, once it reaches the promoter, it binds to the promoter, right? And why? Why is this? What happens? Well, there's a conformational change, right, in the sigma factor. And this causes the double helix. If you look to the right here where I'm drawing, the double helix opens up, right? And this is very important. It has to be opened up so the transcript can be made, right? So now let's look at the next slide here, right? So first we had initiation. The sigma factor binds to the RNA polymerase, slides down double helix until it reaches that promoter. Then that sigma factor binds to the promoter, double helix opens up. And now what's going to happen is that sigma factor is gone. It's gone. And as this happens, well, not as this happens, once it happens, once this sigma factor is gone, there's this formation of a jaw-like structure, as you can see here, that the polymerase creates, right? And the function of this jaw is to hold the ribonucleotide triphosphates in place, right? You need that jaw to hold this in place, right? And then next, in this center section here, you have a rudder. A rudder develops, and that's what separates the double helix, right? And then also you have an RNA exit channel here and an entering. So you have these ribonucleotides entering, and then this is the exit. This is where the newly synthesized transcript comes out of, right? The RNA transcript. Let's see here. DNA single strands reanneal. Okay, so we have initiation, formation of the jaw, and this transcript is being created. Okay. Now we have something called termination. So the previous step was elongation. Initiation, elongation, where the RNA is being created in that exit channel or tunnel. And now you have termination. So that polymerase continues to slide down the DNA until it reaches something called a terminator sequence, right? Here we have it here. We're not going to go into all these different genes and into the specifics, but it reaches this thing. You have promoter here, continues until at the end you have the terminator down here, right? Once that polymerase reaches that terminator, just like the movie The Terminator, what happens is that RNA transcript, it forms a hairpin structure. Why does it do this? It forms this hairpin structure to allow the jaw to open. And once the jaw opens, that transcript is detached and it can go about its life. It could go into the cytoplasm and be translated into the proteins that's needed for the cell, right? So this hairpin structure is very important, as you can see here, and it just tacks off. Boom. So here it is here, and now it's here. So that's very important. It forms that hairpin structure to open up the jaw. So, and that's it for prokaryotic transcription. Very easy, right? Just in 3 easy steps. Initiation, sigma factor binds to polymerase. After that, the sigma factor binds to the promoter, double helix opens up, sigma factor bye, it's gone. Now that the RNA polymerase creates a jaw to hold the ribonucleotides, and then mRNA can be, RNA can be transcribed, right, into a transcript. Then the jaw opens up because of this hairpin structure, and it goes about its business. And that's it. Very simple, very simple. I can do another video on eukaryotic transcription if you guys like. That's it for this video. I mean, very simple. You guys can play it back as much as you guys like. And that's the end of this video. Please leave a like, comment, subscribe. On my channel, you can also check out my website where I have a blog post talking about interesting science stuff, and you guys can discuss it. You guys can disagree with me, agree with me, whatever you guys like. And I look forward to seeing you guys next video, okay?